Welcome to Biology Made Easy. Today we are starting our lessons on mitosis and meiosis, but there are various aspects of biology we need to know before we home in specifically on mitosis and meiosis. So today we will consider the relationship between chromatin and chromosome. Welcome. Chromatin is one DNA molecule called around histone proteins. This chromatin is seen at interphase when the cell is not dividing. Chromosome is the same one DNA that has replicated and held together by centromere, and it's seen during cell division. So chromosome is chromatin replicated, the same DNA. So it's the same one DNA in chromatin and one DNA in chromosome. If this is chromatin and this is chromosome, the two are the same one DNA. This chromatin, it has genes. So I represented only 3 genes with A, B, C. These 3 genes. With this chromosome, is the same DNA that has doubled, and each of the strands is sister chromatids. And it has gene A, gene A, gene B, gene B, gene C, gene C. So it's the same one DNA with the same genes. Chromosomes are seen as tiny as chromatin when the cell is not dividing. So here is a cell at interphase. You have chromatin. Early prophase is chromosomes, but you see them as tiny and look like chromatin. You will see chromosomes as thick, coiled when the cell is dividing. So I have this illustration of chromosome, and I have this as one sister chromatid, and this is also one sister chromatid, and I have gene A on this chromatid. There will be a gene here, gene A. The whole chromosome is full of genes, but I have only 3 genes represented here. Gene A, gene A, gene B, gene B, gene C, gene C. Right? Now, the two are held together by a centromere. Centromere has no genes, and it doesn't pick stain or dyes. There's a telomere at the end of each sister chromatid. Telomere, 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 telomere. The telomere, there are no regions that are genes. The telomeres are just repeating bases. Now, if I have this as a DNA molecule, at the end of it, this black area I call telomere. The telomere is just a cap that protects the end of the DNA. So when DNA is replicating, forming another DNA, the replication goes on through the length, and it just stops at the beginning of a telomere. So the telomere are just caps that protect the ends of the DNA. And these are bases, nitrogenous bases, but they don't form genes. Now, at the end of every cell division, some telomere is lost, cut off and lost. So as telomeres are lost, aging sets in. And with time, as telomeres are seriously lost and some genes are lost, death comes in. So it's been found out that some cells are able to regenerate telomeres. And the cells have enzymes called telomerase. So it's believed that if these enzymes can be made and given as medication, it will increase life of humans. Now, we want to look at the coiling nature of chromatin or chromosome. Well, here is chromatin, DNA with histone. The DNA coils around histone. Right? That is chromatin. The chromosome too, DNA, coils around histone. Now, this is one chromatin and this is chromosome. When it's broken down, it will be seen that this DNA chromatid is coiled, coiled around histone, and that's DNA. Then the same thing, this is a sister chromatid. It's open up, you can see coil around histone. So that is chromatin and chromosome, the same DNA. Now, chromatin exists in two forms, euchromatin and heterochromatin. Euchromatin, the chromatin is loosely coiled, as I have illustrated here, and is seen at interphase when the cell is not dividing. And in the loosely coiled chromatin, the active genes are exposed, they are located in here, so that the genes will produce RNA and a lot of protein for growth. So that is interphase, where growth is taking place. Now, heterochromatin is highly coiled chromatin, and it is greatest coiled at metaphase. And heterochromatin is seen when the cell is dividing. And you can imagine if the chromatin is so much folded like this, the genes that are exposed or located are the genes that are inactive, because all that the cell is concerned at this stage is division, no making of proteins. So the chromosome or chromatid must be highly packed so that as the cell divides, one chromosome or chromatin will not get in the way of another chromatin. Now, there are stains that can commonly stain chromosomes, and it's acetic or propionic ocean or fusin dye. Well, DNA is coiled around histones, not haphazardly at all. Here is DNA, and DNA is coiled around 8 histones. The 8 histones form the nucleosome, and the nucleosome is cylindrical in shape and has these measurements: 11 micromillimeters wide and 6 micromillimeters long. And DNA winds around one nucleosome, making 1 to 10 approximately containing 147 base pairs. And between this folding, there's a linker protein, that's the part exposed, and this linker protein also has 33 nitrogenous base pairs. So you can imagine that when you have euchromatin and is loosely coiled, the active genes will be exposed and be shown the linker DNA isn't. DNA coiled around nucleosomes, highly coiled, very, very much coiled. Well, this is what we have for today. If the lessons have been useful, give us your thumbs and please do subscribe. Thank you very much. Bye-bye.